Good day. Welcome back to studying microbiology and the microscope types, the light microscope specifically. And the last time we studied two types of microscopes, right? The dark field and the light field. No, actually three types. The dark field microscope, the light field microscope, and there was another one. So let me check first if the live is working and then we are starting. Sorry for my voice, and my throat is a bit in bad shape, so my voice is a little bit not clear. Okay. Alright. So, just a bit recap on what we studied last time was the bright field microscope, a little bit of that which remained in the resolution part, and in resolution we studied what the numerical apparatus is and what does the working distance means, what is the theta for, and what does the oil immersion do, and things like that, all of that, formulas and abs numerical apparatus formula, that is d divided by 0.5 lambda divided by n sine theta. This is for the distance. The lesser the distance is, the better we can see the object, means the better the resolution is, or the higher the numerical apparatus is, the better it is for the resolution. And magnification without resolution is nothing. So let's see, in the dark field microscope the background was dark, in the bright field microscope the background was white, or you can say transparent. While in phase contrast microscope, let's see what happens. Unpigmented living cells are not clearly visible in the bright field microscope because there is a little difference in contrast between cells and water. They are saying that if in bright microscope we don't stain the cells, living cells like bacteria and fungi, etc. If we don't stain them, or if they don't have pigment in them naturally, they are not clearly visible and under the bright field microscope, because like in chloroplast there will be not chloroplast, like in the cyanobacteria for example, there will be chloroplast and that is pigmented green, so they will be looking a little bit more visible under the bright field microscope, but it is not the case for all of the living cells. So I think for that case they developed a phase contrast microscope. One solution to this problem is to kill and stain cells before observation to increase the contrast and create variation in the color between cell structure. So they did say to kill the cells. What is the need for killing the cell? Usually when you kill a cell, the cell loses its water content, some kind of, or there is a physiological change occur, and that because of that they retain the stain more better than the living cells. Also, living cells are less visible than the stained cell. They are more visible even if we don't stain them. So they are saying that in phase contrast microscopy we kill the cells and we stain them, we color the cells for observation to increase the contrast and create variation in the color between cell structure. So basically it's just your typical type of microscope where we stain cells and then we study under microscope, just like we used to do in our lower classes, right? But what if an investigator must view living cells in order to observe a dynamic process such as movement of phagocytosis? Like they are saying, what if there's an investigator and they want to study the living cell as it is, like how do they do phagocytosis, such as phagocytosis means eating, process of eating, right, by cell. Phase contrast microscopy can be used in this situation. A phase contrast microscope converts these slight differences in the refractive index and the cell density into easily detectable variations in the light intensity and is an excellent way to observe living cells. So phase contrast microscope convert the slight differences in the refractive index. Of course, there are changes in the or differences in the refractive index, and I think they are talking about the refractive index of the lens or the medium in which it is present. They are saying that they convert the refractive index a little bit and the cell density into easily detected variations in the light intensity. So they change the refractive index and they change the cell density, and that make it easier for detection in the light intensity, like in the light microscope. And they are saying it is an excellent way to observe living cells. I still don't know how they change the refractive index, which is going to be explained below. I think so. And undeviated waves will be about 1/2 wavelengths out of the phase. Oh, so they change the wavelength of the light as well. Undeviated waves. Undeviated means that the waves that are not reflected or not refracted, like the ones that straightly go into the lens, right, will be about 1/2 wavelength out of phase. Like they will change the wavelength of the light by half of the portion. So if the wavelength is 400 nanometer, I think they will change it to 200 nanometer or something, and will cancel each other when they come together to form an image. What is this? Undeviated waves will be about 1/2 wavelength out of phase and will cancel each other when they come together to form an image. The background formed by undeviated light is bright, while the unstained object appears dark and well defined. This type of microscopy is called dark phase contrast microscopy. I didn't get what they were saying. Let's see if we can understand it by diagram. Annular stop. Annular stop. What is this? Alright, so in this case the light is coming from the sides, like in a circular shape, like it was in the dark field microscopy, right? And then there is a condenser. So it's for up till this nothing changed. I think the change happening here. So diffracted rays are retarded 1/4 length after passing through an object. So when a light passes through an object, or you can say wavelength in this case, if it is diffracted, it is changed to 1/4 of the length. Direct light rays are advanced 1/4 wavelength as they pass through the phase ring. Direct light rays are advanced 1/4 length as they pass through the phase ring. Most diffracted rays of the light pass through the phase plate unchanged because they miss the phase ring. Most diffracted rays of light passes through the phase plate unchanged because they miss the phase ring. Most diffracted rays of light passes through the phase plate unchanged. Diffracted rays are retarded 1/4 of the length after passing. Direct light rays are advanced 1/4 length. Most diffracted rays miss the phase ring. Oh, so this is diffracted ray, and they are changed to 1/4 of their wavelength. And when they are passing through it, this is the phase ring. Here and here is 2 rings, but the diffracted rays miss it. Then what happens? Amplitude contrast is produced by the light rays that are in reverse phase. In the reverse phase. dark image with a brighter background. It is not clear yet. What is this? Oh, I think I missed the part from here. A phase contrast microscope converts slightly differences in the refractive index and cell density into easily detected variation in light intensity, and is an excellent way to observe the specimen or cells. The condenser of the phase contrast microscope has an annular stop, an opaque disc with a thin transparent ring which produces a hollow cone of light. Alright, so the condenser of the dark field microscope didn't have this annular stop. It had the opaque stop, like which was completely blocking the light. And this is also an opaque disc with a thin transparent ring, but it has the addition of a transparent ring, thin transparent ring, which produces a hollow cone of light. Alright, so in that case, the light was going from the sides and there was an opaque object in the middle of it. While in this case, the ring is in the opaque object and the light only goes through the transparent ring, making a hollow cone of light. And as this cone passes through the cell, some light rays are bent due to the variation in the density and refractive index within the specimen and are retarded by about 1/4 of the length. Alright, so it's a natural process. When it passes through the specimen, of course, the density in the cell is different in one area or in another area, and the changes to 1/4 length are retarded by about 1/4 length, are changed by about, or are they? I think they are saying like 1/4, like total wavelength. The fourth part of it is, or you can say 1/4 part of the whole wavelength is retarded. So the deviated light is focused to form an image of the object. So the light that is deviated or that is changed, undeviated light rays strike a phase ring in the phase plate, a special optical disc located in the objective, while the deviated rays miss the ring and pass through the rest of the plate. Alright, so there are two kinds of light for us for now. One is the deviated light and one is the undeviated light. The deviated light is used to form image, while the undeviated light pass through the phase ring in the phase plate, right? And they are saying that the deviated light don't pass through this phase ring in the phase plate, miss the ring and pass through the rest of the plate. It is because their wavelength is changed. They set the phase ring in such a state where they know what kind of wavelength will pass through this phase ring, right? But when the cell density is different in the specimens, their wavelength are changed out to about retarded by about 1/4 of the wavelength, total wavelength. So they don't pass through that phase ring. Yeah, so they don't pass through it. Because they are deviated, they are changed, they are not their original wavelength. And they pass to the rest of the plate, right? Like they don't pass to the ring, but of course where else will they go if they don't pass to the remaining area? So this is the ring and this is the ring, while these are the remaining parts of the face plate, right? So here there is a light. Now there are two types of light for us, right? The wavelength here is all the same, right? Is all the same. But when it passes through the specimen, its wavelength changes by 1/4. If it don't pass through it, if it is not deviated, the wavelength remains the same, right? Same. Now if the one that isn't changed or is not deviated, they pass through the phase ring, while the one that is deviated doesn't pass through phase ring, while it goes into the phase plate other than the phase ring. And they are saying that the deviated light is used to form the image. Now let's see what else happens. If the phase ring is constructed in such a way that undeviated light passing through it is advanced by 1/4 wavelength, then the deviated light and undeviated lights will be about 1/2 wavelength out of phase and will cancel each other when they come together to form an image. Alright, I think I'm getting a little bit. So guys, here the deviation was by about 1/4, right? And they are saying when they passes through this phase, they only capture 1/4 wavelength advancement, right? Changes, they know the original one. Or the phasing changes the wavelength by 1/4, right? So now when they pass through, this one is plus 1/4, this one is minus 1/4, right? So their changes is like about 1/2 wavelength from one another. If you see the difference, like 1/4, like 1/4 part of this and 1/4 part of that, and if you remove that, there is only 1/2 difference, right? So when they pass through it and comes together, of course the plus and minus will cancel each other and they will form an image. After that, of course, they are going to combine with one another, because in the starting the wavelength wasn't a different wavelength, right? So of course when they are going to combine, they want to go to the original position, but in this case they can't, because the wavelength has been changed to, like you can say, about 1/4 up and down, and here this becomes 1/2. The background formed by undeviated light is bright, while the unstained object appears dark and well defined. So they are saying the background formed by undeviated light is bright, while unstained object appears dark and well defined. This type of microscopy is called dark phase contrast microscopy. I kind of get the gist of it, but I don't think I'm completely clear with it. Amplitude contrast is produced by the light rays that are in reverse phase. Reverse phase. Guys, shouldn't we search the, wait, we should search what is the retardation of wavelength. I think we'll get from here. The difference in the optical path length experienced by two polarized light waves when passing through a bifringent material. How much one wave is delayed compared to the other, expressed as a fraction of wave. Oh, I see. Oh, guys, I get it. Those people who excel in the physics, I think they wouldn't have taken that much time like me. Alright, let me show you by what they mean it, right? Like, I completely get it now. Let me show you. Here. Guys, take if this line, right? This line and this line. Now there is a wavelength. Consider this as a 0, right? This is like normal point of, right? Now there is a wavelength that is passing through at normal, right? So cut this wavelength into 4 parts, right? Crust, like you can say this, what? Should we start from the middle point? Yeah, we should start. So we will end in the middle point as well. So this is a complete wavelength, right? This is a complete wavelength for us, right? Now we cut into 4 parts like this. A complete wavelength is like this. So 1, 2, 3, 4. We have, forget if my wavelength is not, completely okay with the distance, but all of these things are equal. So this is in the 4 parts, right? Like 1/4, 2/4, 3/4, and 4/4, right? So if you look at this, by the way they means that it is retarded by 1 by 4 means, let's consider retarded means 1 minus 1 by 4, right? And they say advanced by 1 by 4. Let's say if it is plus 1 by 4, right? Now they are saying that the plus 1 by 4 wave will move like a little bit faster, or you can say it will be a little bit more in the future than this wave, and this one will be a little bit delayed than this one, like in the past. For example, you can say if this is advanced, that means that this point should already be here, like, right? Wait, shouldn't we go below this? Right, like there it is starting already now. That means that by the time a normal wavelength will be passing through 0, this plus 1 by 4 wavelength will be passing through this point, and it is already advanced by 1 by 4 part. This 1 by 4 part is of another wavelength, while in minus 1 by 4 part this part will be delayed in the past, like it will be here somewhere, right? So this part, this 1 by 4 part is still here in the past, like it is retarded, it is delayed, while this one is advanced. So plus 1 by 4 means the wavelength is advanced, while in minus 1 by 4 you can say retarded wavelength, that means it is not advanced. So when so when they mean that when these wavelengths, for example, this is minus 1 by 4 and this is plus 1 by 4, this is the change in their wavelength, right? This is the change in their wavelength, like this one is in the past, this one is in the future. So when they combine, what will happen? They will cancel one another and they will bring back everything to their original position, like this original wavelength, they will bring it back. How will they bring it back? Just think about this. If you overlap this one wavelength over another, the thing that is passed already and the thing that is delayed will cancel one another, and the wavelength will come into its original shape, and then we will get the image. Although I get this point, but I don't get why they are going through the trouble of changing the wavelengths. And here this reverse phase is the same thing, like it is, backwards. So I still don't get it why they are going through the trouble of changing their wavelengths by, like, you can say 1 by 4 advancement and delay to produce an image. Like, what does this dilation and advancement actually do if they are going to cancel one another in the end? Will be about 1 by 2 wavelength out of phase. Now where is 1 by 2 coming from? Will they not cancel one another? Okay, well, let's see what they show in this diagram. This is a bacterial cell, and two kinds of waves pass through this. Two kinds of waves. So radially deviated by specimen is 1 by 4 wavelength out of phase, right? This is in the past. So the deviation will be 1 by 4. The deviation is 1 by 2 wavelength out of phase. Here the deviated ray was 1 by 4 wavelength out of phase. There this is 1 by 2 out of phase. Why did they deviate it completely 1 by 2 out of phase? Didn't they say that the 1 by 4 wavelength advance, like it will be advanced by only 1 by 4, but here they are saying that it will be advanced by, you can say, 1 by 2. Alright, if we change that deviated and undeviated like this, like we studied already, 1, 2, you can say 4 parts. And here another 1, 2, 3, 4. 1, 2, 3, and 4. So when it was passing through the specimen, it was delayed till this much. Means that line that is in the center came here, right? But when it passes through this phase plate, it retarded it even more. It deviated it even more into even 1/4 part again. So this half part is delayed. And this half part, you can say it is advanced. This wave is like this. Well, this one will be like this, right? Wait, Control plus Z. Right. So if you look at this wave in comparison to that wave, it is in the future. There its trough is looking, there it is trough, and there, no, this is crust and this is trough. So when this happens, when a wave has crust and trough across, or you can say against each other, they cancel out one another because their energies, you can say, cancel out one another, because this is in their lowest position while this one is in their highest position, and they cancel out one another. When they cancel out one another, they are saying that this will create an image. I see what they meant by 1/4 wavelength advancement. Like they like to play with their words. They could just have said that it is deviated even more by 1/4 and it became 1/2. No, but they have to say that this one is first deviated 1/4. That means that at this stage it is already deviated 1/4 wavelength, and this one is already advanced 1/4 wavelength. Right, not from here. Like in this case, if it is deviated 1/4, that means it is already advanced by 1/4. But when it passes through that, if that is going to go 1/4 advanced, then this one is going to go 1/4 deviated. So in total, that one is 1/2 advanced wavelength, while this one is 1/2 out of phase, or you can say retarded wavelength. So when they come across each other, they cancel out one another in the lens, because the lens focuses those two at the center point. And here they cannot cancel one another because they are not crossing one another directly, while in the lens they do cross one another directly at a center point, at a focal point. I see. Right. Now what happens? Color filters often used to improve the image. Okay. Phase contrast microscopy is especially useful for studying microbial motility, determining the shape of living cells, and detecting the bacterial components such as endospores and inclusion bodies that contain poly-B hydroxyalkanoids, alkane OH, acids, alkanes. Hydroxy is OH. Is OH alcohol? Wait, isn't it OH? I'm thinking way too much. So they are saying it is useful for studying the microbial motility, like how my microbial cells are alive and how much are not, because the density differs. The density becomes higher when the cell dies. Determining the shape of living cells. Yes, shape of living cell also matters because every living cell shape deviates the light in a different way, or you can say in a different pattern. Detecting the bacterial components such as endospores. So because of that, you can, because it only, rely on pattern instead of just the light capturing as well. So we can determine the bacterial components such as endospores and inclusion bodies. Inclusion bodies are the abnormal bodies or unnatural bodies that are present in microorganisms due to some other reason. Like bacteria, or you can say cell normally don't have these bodies. But because of infection by some other organism or attacked by another component from the environment or something, the inclusion bodies becomes, and sometimes inclusion body is not harmful, but in human cases when the virus attacks them, the inclusion bodies are harmful, or you can say it determines that the cell is attacked by a virus. So they, in this case, they are giving example of the inclusion bodies that contain poly B hydroxyalkanoates, for example poly B hydroxybutyrate, polymethyphosphate, sulfur, or other substances. These are clearly visible because they have refractive indices markedly different from that out of water. You're right, like they are just comparing them to water. As long as it is different from water, they will be visible. Like their focus is just to get a refractive index that is different from water. So phase contrast microscopy also are widely used in studying the eukaryotic cells. Wait, let me see the diagrams. So this is Pseudomonas disulfatomaculum. What is this? This is a protozoa, isn't it? Wait, let me go search it in a Google. Again I am doing this. Oh, it's a bacteria. I thought it was a protozoa. When did bacteria started looking like this? I see, so it's a bacillus. It's not like a V end shaped bacteria. Their ends are actually quite pointy, which is not commonly in bacillus, but maybe this shape is just looking like this. Usually, yeah, all of them are round. You can say those two are combined. I think they are not the chained bacillus, they are separate bacillus. And these are endospores, I think, the ones that are shown in the sulfatomaculum bacteria. Wait, does desulfometaculum bacteria have or produce spores? What if I'm wrong? Oh yeah, so they do produce spores. And permeation phase contrast microscopy. Again, the background is dark, and no, it is. Wait, didn't we said that the background should be bright? What do they mean by bright actually? This is blue, this is green, and this is blue as well. Didn't they mean transparent? What does bright mean in phase? Yeah, phase contrast microscope. Areas of a specimen that appears lighter or more illuminated compared to the surrounding background. Oh, so they actually means bright. Bright. I thought bright was more like invisible, or you can say transparent. But here they means that it becomes lighter than the one, or you can say more. Like the more brighter you are, the lighter you will get. The less brighter you are, the darker you are getting it. So the background is brighter, so it will have lighter color as compared to the organisms. I get the process how it works, but I still don't know how changing the wavelength will make you or give you, especially after cancelling out the background and things like that, to give you a background differently. And I have studied this whole chapter about lenses and things, and this is making it even more confusing as to how the lenses will. How does this light create an image? Do I take a class of how the eyes work? Otherwise, I'm not getting how when these two combine and becomes a complex and make a straight line will create an image. I think I do need to study the eyes working, or you can say how we see the images through the light class separately. Phase contrast, alright, we are done. So, endospores and inclusion bodies. 2.8D. Oh, so these are inclusion bodies. 2.8. This is 2.8, while this is D. Oh yeah, here it is explained. A phase contrast micrograph of the Pseudomonas cell, which ranges from 1 to 3 micrometer in length. Desulfotomaculum acetoxidans with the endospores phase contrast. Alright, I was right. These were endospores. And this is paramecium stain to show a large central macromolecule with a small spherical micronucleus at its side. Phase contrast microscopy. Alright, so the denser the object is, the darker it will look. Like if you look at the background, it looks less bright, no, more bright, because the deviation didn't occur that much. If you look at this diagram, it is denser and deviation occurred, so it looks darker. But in this case, the density is even higher than the cell, like the micronucleus, the macronucleus. So it becomes even more darker. We can see it more clearly. So phase contrast microscope. The denser you are, the higher density you have, the more darker you will be looking in the microscope. Right, so the differential interference contrast microscope. The differential interference contrast microscope, DIC microscope, is similar to the phase contrast microscope in that it creates an image by detecting differences in the refractive indices and thickness. They are saying for now it works the same because it studies or it detects the differences in the refractive indices, and thickness by thickness they means the density, right? Two beams of the plane polarized light are at right angle to each other, are generated by prisms. I see, it looks like they changed the lightning source somehow differently in the interference contrast. So they are saying two beams of plane polarized light. Polarized light means there is only one wavelength. In white light there is multiple wavelengths, while in polarized light there is only one wavelength. And the wavelength, we know which wavelength is. Like for example, for the wavelength of around 400, you can say 500. Is it blue or green or red, whatever? And they are saying we have two beams of them and they are at right angle to each other. Right angle. One beam of light, another beam of light. They are at right angle to each other. And this is generated by prism. We know prisms usually split the white light, so it can maybe used to produce polarized light. So in one design, the object beam passes through the specimen, while the reference beam passes through a clear area of this light. After passing through the specimen, the two beams are combined and interfere with each other to form an image. A live unstained specimen appears brightly colored and three-dimensional. Structures such as cell walls, endospores, granules, vacuoles, and eukaryotic nuclei are clearly visible. Alright, so this works by comparison. Didn't they show diagrams somewhere? They haven't shown macro diagrams. We will see it in the Google anyway. So in this case it works by, there are two beams of light and they are at right angle to one another. One light is passed to the specimen, and this is polarized light. Remember, it is a polarized light, means this is only one wavelength, and it is better to have one wavelength because we know how the wavelength changes occur than more clearly than when you have more wavelengths to deal with. One wavelength is passed through the specimen, one wavelength is passed through a clear area where there is no specimen. And then when we combine that two together, it forms a more three-dimensional image and can give us result more brighter and more clear. So it's like we're comparing them to one another and combining them. Like this is the area without the specimen, and here this is the area with specimen, and we combine them to make a more complex image. Let's go how this DIC microscope actually looks like or works like. DIC microscope construction. Will that be fine? Specimen condenser, objective specific prism. So this is prism, and it changed. Oh, I see what they were doing with this. So they did polarize light. Where is the right angle here? These are parallel. Parallel don't have the right angle. I want to see how the right angle works here. This is the polarizer. 45 degree. Beam splitting. Oh, here. The 90 degrees. I see, so that 90 degrees happens inside the prism. Where did it go? Here. Like I can't enlarge it more than this. So if you look at this clearly, doesn't this look like a 90 degree? Almost 90 degree. And this happens in the prism that it becomes, no, it has to be exact 90 degrees. Why will I compromise on the angle? Why haven't they write it? Wait. Prism splitting at 90 degrees. Let's see if we get something better. Who calls this a 90 degree? They brought out the most complex pictures I have seen of the microscopes, and they don't give us the answer. Ordinary ray, extraordinary ray. Right angle prism, ray extension. What is this? Right angle prism. Oh, wait, was the prism right angle? Was I reading it wrong? Wait. Two beams of plane polarized light at right angles to each other are generated by prisms. This happens. I kind of get it. I don't need more explanation, light. So this is a prism. If you see here, I think you can see the light generating as, like this diagram didn't show it correctly. There was another diagram that has shown that the prism split the light at the 90 angles. And inside the prism it changes a little bit here and there. Well, prisms are of different types, you don't know. And the wavelength, became at right angle, you can say. Yeah, right angle is 90 degree. Why is there already 45? and then when it splits, it passes through different areas, one through specimen and one doesn't go through the specimen, right? And then it combines in the lens, and then it creates an image, right? Oh, so there is another prism as well. So one prism is for splitting and one prism is for collecting. They didn't mention this part. Oh yeah, so they are talking about only one design. Yeah, they didn't mention how the two beams combine and interfere with each other to form an image. Right? Guys, the more we go deeper, the more we will find more things, and I don't think we need to go inside that. We'll do some casual live and study how the eyes work, how our eyes can see the light, right? So the fluorescence of microscope. This is another, wait, what did I say just now? The fluorescence microscope, and by name it shows that there will be some fluorescence used in the microscope. Like for there, it was just the shadow, it was just the image, right? Just the image. While there we will detect with lightning where the things are present, where not. Let's go into the detail of the fluorescence microscope. The microscope thus far considered produce an image from a light that passes through the specimen, right? The microscope that we studied till now, we considered that they produce image from the light that passes through the specimen, right? This is how it happens normally. The light passes through the specimen and then it is collected, or you can say changed by the lenses and combined or whatever done. Then we see it. An object also can be seen because it actually emits light, and this is the basis of the fluorescence microscopy. Alright. They say we don't need microscope because the object emits light as well. Like if you look at the wall, it is emitting light. That's why we are able to see the wall, right? Usually this happens. But the small microorganisms, they emit light as well, but we are not able to see them because they are way too small for us to, separate them into different objects. So that's why in the previous cases we were enhancing the resolution of the microscope to see them differently by different methods. But in this case they are saying we don't have to separate them like that. We will utilize their emission of light as a base. So this is the base, that all the objects emits light, right? We see the object because it emits the light, and this is the basis of fluorescence microscopy. So when some molecules absorbs radiant energy, they become excited and later release much of their trapped energy as light. Right. Some molecules absorb energy, they become excited, and when they want to go back from that excitation phase to the lower energy phase, they release that energy. So any light emitted by an excited molecule will have a longer wavelength or be of lower energy than the radiation originally absorbed. Why does that happen? Always remember that the light absorbed have more energy than the light released, or you can say energy, packet of energy. Light do have energy. The higher the wavelength, the longer the wavelength is, means the lower the energy. The smaller the wavelength is, the higher the energy is. So they are saying that when light is emitted, or you can say given out, in given out form it is in the lower form of energy, that is longer wavelength. It doesn't mean necessarily that it has to be the longest wavelength. It just means it becomes longer than what we originally provided them with. Fluorescent light is emitted very quickly by the excited molecule as it gives up its trapped energy and returns to a more stable state. They are saying fluorescent light is emitted very quickly by the excited molecule as it gives up trapped energy and returns to more stable state. In chemistry, we always say that molecules strives to go into a more stable state, right? This is usually a case for all of the molecules, objects, and whatever. They want to go to more stable state. They do take that energy, but then they think, yeah, that we don't need them. This is not our stable state. And usually in chemistry we study the lesser energy you have, the more stable you are. And so there are some molecules that release that energy quickly, and there are some that don't release it quickly. But in fluorescent light, it is emitted very quickly for them to go back to more stable state. So the fluorescence microscope exposes a specimen to ultraviolet, violet or blue light and forms an image of the object with resulting fluorescent light. Like usually the objects receive white light, right? But in fluorescence microscope we give them the light that we want to give them, and it is of high energy. Remember, white light have different types of energies, right? It isn't of one wavelength. It has a lot more wavelengths in them. So you have to, make them one wavelength for you to study it easily. So in this case, violet or blue light has the lowest wavelength and the highest energy, or you can give them ultraviolet because it is even smaller wavelengths than the violet or blue light. It is out of the visible range of light and it has higher energy, so it forms the image because the objects take that energy and instantly release it back, and then the released light that is reflected, we capture them and we form an image out of it. The most commonly used fluorescence microscopy is epifluorescence microscopy, also called incident light or reflected light fluorescence microscopy. All epi means up, above, right? This is the word of some other language, I don't know, German, whatever. So epifluorescence means you put the light upon something, above something. And incident mean as well that thing, like the thing that is being incident upon something, put upon something, comes in contact with something. So incident light is the coming light or the reflected light. So you can say it happened because you put the light on them of your own choice. It's the same thing as you can say that you study the reflected light because it emitted the light that you incident them upon. So it is the epifluorescence. In all this sense, you put the light that you wanted of your wavelength of the higher energy and they release it back. This is the basic principle. In this case, we don't study the light passing through, like we don't study refraction. We study reflection. Refraction. Till now we were studying refraction, right? But now we are studying reflection. Right. Refraction, till now we were studying this. Right? But now we are studying this. This is what we are studying now. So this is actually cool. I didn't know that we can use like things like this as well. Right. So epifluorescence microscope employ an objective lens that also acts as a condenser. Right. So all of that light that is reflected, you have to sort it out. So in this case, there is an objective lens, but that also act as a condenser. Usually objective lenses, or you can say lenses, that are usually convex. We use convex lens in the microscope, and it usually works to bring out the light to a focal point, right? A middle point, to focus it all in one place, to project it on one place. But they say in this case it acts as a condenser as well. Condenser means, of course, they condense all the light that is splattered around here and there, right? Because it is reflected light. The reflected light don't know where I'm going. It only reflects because of the density of the object. The more denser the object is, the more the reflection will be, drastic. Alright. A mercury vapor arc lamp or other source produces an intense beam of light that passes through an exciter filter. A mercury vapor arc lamp or other source produces an intense beam of light that passes through the Exciter filter. Oh, vapor arc lamp. Why do we use mercury vapor arc lamp? Wait. Vapor mercury arc lamp. What is this thing? Mercury vapor arc lamp. Sorry for my spellings. I don't want to bother with it. Oh, so there are mercury vapors in there instead of gas, some kind of gas. Yeah, and so the filament in the light break down that vapors and produce ions and produce light. But why use mercury? So it produces the intense beam of light, high intensity light. Right here you can see the electric arc that mercury vapor creates a bright output. Okay, so this is how it works. Wait, I forgot where I was. An exciter filter. The exciter filter transmits only the desired wavelength of the excitation light. Right. Like we knew that this is going to be only one wavelength. But when mercury vapor arc lamp produce a light, it is not of one wavelength. It is of multiple wavelengths. So when it goes to the exciter filter, it only allows only one type of wavelength to pass through it and not the all types. The excitation light is directed towards the microscope by a special mirror called as dichromatic mirror. This mirror reflects the light of the shorter wavelengths. That is excitation light. Right, like you will be thinking, if you put a mirror, there is light in the environment, right? There is light in the environment. What if it reflects that light as well? No, there is only a dichromatic mirror, a specific type of mirror, and it reflects only smaller wavelengths. So it means that the white light in our environment is not reflected by it. Dichromatic mirrors. Different will, huh? What? Dichroic? I did write a dichromatic. Alhamdulillah. Guys, if you didn't notice, I am not feeling well, so that's why my voice and yeah, my nose is running. But I didn't want to miss this book. It's fun studying. Dichromatic. So I did search for dichromatic mirror, but they are bringing out the a mirror that reflects light of a certain wavelength. Wait, let me look at the images. Oh. This is dichroic filter. Mirror, sorry. Oh, so this reflects the light. From which angle does this look like a mirror? Mirrors are usually silver. What is this? Aren't mirrors supposed to be silver looking? Yeah, this is how mirrors look. What was that about? Oh my goodness. They appear to be whatever they are reflecting. Whatever. So there are mirrors that looks a little bit different. But what if we look at this diagram? It didn't look like it was reflecting. It looked like it was refracting. Look at this. Look at this. From where does this look like or is this a mirror? From where does this look like a mirror that is doing a reflection process? This looks like doing a refraction process. Wait. Here. This is a light source. This is a dichromatic filter. This is the same sample. Light source, sample, light source. What is this? Dichromatic mirror. This is a light source. This is excitation filter. This is a dichromatic mirror. Alright. So this is how normally works. But instead of this bulb, think of it as a, where did it go? Oh, it is gone. Whatever the light was coming, it passes through the excitation filter to filter out only one wavelength, and the dichromatic filter passes through and filter it even more to only one short wavelength. That is the excitation light. Alright. Oh guys, look at this first. Differential interface contrast microscopy. A micrograph of protozoan amoeba proteus, a three-dimensional image contains considerable details and is artificially colored. Oh, I don't believe this is done by differential interference contrast media. Look how 3D it looks, just as it talked about it. So, to excitation, the light continues down, passing through the, alright. So about the dichromatic mirrors, it looks like they are the types of mirror that reflects only the shorter wavelength light, and they act as a refractor. They allow the darker wavelength light pass through them. So it acts like a refractor for the longer wavelength and as a reflector for the shorter wavelengths. This is cool. So the excitation light continues down, passing through the objective lens to the specimen, which is usually stayed with a special dye molecule called as fluorochromes. The fluorochrome absorb light energy from the alright. So fluorochromes, the stains are done so it makes it easier for the sample to absorb light, right? Usually it absorbs a normal amount of light, and because of that normal amount of light we weren't able to see them before, right? But now in microscope we stain them with fluorochromes, and because of that it absorbs more light, more fluorescence, and it appears even more brighter than before, and because of that we are able to enhance the resolution of the microscope and we can see it. So the fluorochrome absorbs the light energy from the excitation light and fluorescence brightly. The emitted fluorescent light travels up to the objective lens into the microscope, alright, because the emitted fluorescent light has a longer wavelength and it passes through a dichromatic mirror to a barrier filter which blocks out any residual excitation light. Yeah, so they are saying once the fluorescence light is emitted, means this is the second step of the fluorescence. The absorption process is done. Now is the emission process. So they are saying the emitted fluorescence light has a longer wavelength. We know it's a longer wavelength. So when it is reflected back to that mirror, it isn't reflected. It is passed through it because it is of longer wavelength. And it doesn't act as a barrier. It only acts as a barrier for the shorter wavelengths. So in this case, it kind of acts as a filter. Like it only allows the the longer wavelength light to pass through them. So it blocks out any residential excitation light. Finally, the emitted light passes through the barrier filter to the eyepiece. Alright, it goes to a further filter, and that is barrier filter, and to the eyepiece. The fluorescence microscope has become an essential tool. We don't have to study this before going through this. We look at this diagram. So this is our sample. This is covered with fluorochrome. This is mercury arc lamp where the light is generated. This is the exciter filter. It removes the longer wavelength because we don't need them. And only the shorter wavelengths pass through it, while this dichromatic mirror reflects short wavelengths, transmits longer wavelength, means allows the longer pass. So for now we had only shorter wavelengths. The waves that are coming from here are only the shorter wavelengths because we don't want to remove this exciter filter, because then longer wavelength lights will pass through this and it will go here and it will change our image structure or things like that. So that's why we put the exciter filter. So it only allows the shorter wavelength which is reflected to the sample, and sample absorbs that light, and then the removes, or you can say emits, the longer wavelength. The longer wavelength is shown by the red arrow, and it is there is a barrier filter, and there you can see is a lens, objective lens. Where is another objective lens? And so it acts as a condenser as well, and so we can see the picture image. Actually, this microscope is way easier, way more easier than the previous microscopes. Like, can't we make it at home? Like, we just need a dichromatic filter, oh yeah, mercury lamp, and fluorochromes. Yeah, isn't this enough? Like, this is pretty simple than all of the other microscope. I wonder what is the pros and cons of all of these types of microscope. So the fluorescence microscope has become an essential tool. Like, does it have any drawbacks in the medical microbiology and the microbial ecology? Alright. Bacterial pathogens, for example, Mycobacterium tuberculosis, the cause of tuberculosis, can be identified after staining them with fluorochromes or specifically labeling them with a fluorescent antibodies using immunofluorescence procedures. Guys, this other part isn't related to the microscope, alright? This is the fluorescence antibodies, and this is the serological testing, and you don't have to know this because this is, well, we are going to study in the future chapters, but this isn't related to microscope. In ecological studies, the fluorescence microscope is used to observe microorganisms stained with the fluorochrome labeled probes, or the fluorochromes that bind specific cell constituents. So they are saying in ecological studies, right? In the environmental studies, the fluorescence microscope is used to observe what microorganisms? Probes. Probes, guys. Probes are the part of the DNA, right? But it is fluorochrome labeled. Like, it does absorb and emits light. So fluorochrome labeled probes, actually we don't study these probes. We use these probes to attach to some parts of the cells or some organelles or things like that, and then we can see them under microscope if we have the desired object here or not. Or you can say, yeah, here it is written. Or you can just say the fluorochrome. Use just simply the fluorochromes. You don't have to use the fluorochrome labeled probes. Like, you don't have to go the one step further, but there are some objects, or you can say some cells, that cannot be, fluorochromed, staining doesn't work on them. So in that case, maybe because of that, we use the probes like DNA particles to attach to them. Like we indirectly attach the fluorochrome particle to the cells. So in addition, the microbial ecologists use epifluorescence microscopy to visualize photosynthetic microbes, as their pigments naturally fluorescence when excited by light specific wavelengths. Alright, so they are saying we use epifluorescence microscopy even if don't use the fluorochromes. Their natural pigments in them are actually produce fluorescence, right, of specific wavelengths. So it is even possible to distinguish live bacteria from dead bacteria by maybe this kind of microscope. Oh, by the color they fluorescence after treatment with a special mixture of stains. Right. If you right here in the diagram, like they are saying, if you color two types of cells with the same color, but one is dead, one is alive, both are going to take the light differently, right? Like for example, in the first one, they dyed the live and dead organisms. So dyes that cause live cell to fluorescence greens and dead ones red. So there is a dye, right? There is a dye. They colored them. It was only one dye, and they colored them for both living cells and dead cells. So the light that was produced by living cells was green. Like if you see here in the sample, the green ones are the living cells, while the dead ones are the red ones. I don't know what type of sample they were collecting, that there were half of them dead. So they are saying that this is actually pretty fascinating, right? That we can separate the dead and living bacteria or microbes just because of their fluorescence. But we have to apply a specific dye before that. Thus, microorganisms can be used and directly counted in a relatively undisturbed ecological niche. So they are saying the microorganisms can be viewed and counted. Like you can see, you can count it pretty much easily than any other type of staining you will do in a relatively undisturbed ecological niche, like without disturbing them, like without having to go through a long procedures and things like that, without disturbing their functioning, etc. You just color them and like nothing changes. Aramine is used to, like you don't have to use alcohol, like for example in gram staining, where we differentiate between gram positive and gram negative bacteria. Like when we do gram staining, we do iodine passing, alcohol passing, like things like that, and I think that disturbs their natural functioning. So in this next diagram, aramine is used to stain Mycobacterium species in a modification of the acid phase technique. Alright, so in this case is Mycobacterium species. Like we wanted to know if these bacteria are Mycobacterium species or not. So we use a stain that is aramine, right? And this is the modification of acid phase technique. Guys, the acid phase staining is coming, in the future here, staining of species, etc. So we will study that in the future. But they are saying that here aramine is used instead of you can say in modification to this technique. Fluorescence antibodies tag specific molecules, just as we studied about probes, right? Like the DNA particles, we do use antibodies as well, and we attach some fluorescent molecules to their antibodies, and that antibodies go and bind to the organism or molecule we wanting to see. Usually this is serological, but in this case this is attached to a microorganism, so we can study it under the microscope. So in this case, the antibody binds to a molecule that is unique to the Streptococcus pyogenes. Like in the diagram you are showing is of Streptococcus pyogenes, and we can see it only because the antibody, fluorescent antibody, is attached to it. And all of these diagrams are 10 micrometer. So this is it for the types of microscopes. And guys, I was so excited about this topic that I wanted to skip not the microscope topic, I'm talking about the staining topic. That I wanted to skip the microscope part, right? So we are going to go to the staining part tomorrow. No, I skip one day and I do that. Right, this is the most interesting, no, not interesting if you ask me, well, interesting, important, important aspect of microbiology, the staining techniques, right? Without staining we can't do microscopy well. We need to stain. We need to stain it. Like staining is coming first. Microscope may be advanced as much as you want, but if you don't do staining properly, you can differentiate between microorganisms, and the microorganisms are, so picky. They want different types of stains for different types of microorganisms. Fungi has their type of staining, bacteria has their type of staining. Like, different types of dyes, stains are used, basic dyes, basic, acidic, normal, neutral, acid based, acid phase, and things like all of these kinds are, we are going to cover that. And also we don't just stain the living cells, we do stain the capsules, and extra outer covering around the bacteria, after the cell wall. And endospore staining, we do that as well, because endospores are actually identify, we identify organisms presence because of their endospore. If we know whose endospore is this, we know what type of bacteria infected here, or what kind of bacteria will grow out of this endospores. We do flagella staining as well, because fungi, or you can say bacteria, usually have categories because of their flagella. Like some bacteria has one flagella, some has 2, 3, 4, some has on the outer side, some has covering it around their body, and things like that. Transmission on, yeah, in this, the electron microscope is going to go into a next chapter, or into a next live, right? So I did study types of the microscope, specifically the light microscope, but this light part, I think we are a bit less knowledgeable on that part. So I'm thinking of studying how our eyes work, right? How our eyes can see things by light, through light. Like what is it actually in our eyes that, or how actually is the image produced by these lights, right? Like I want to go deeper in that aspect. So yeah, this was it for today. I don't want to drag it in even more, and stay tuned for the next staining techniques, because it is way too important for you to skip it. This is the end. Goodbye.